Here in part 3, let's explore hypotheses for the origins of eukaryotes. This is an expanding area where we're making discoveries virtually every year. But we believe, based on the currently available data, that the data are consistent with eukarya and archaea forming one branch of the tree of life, with this branch here being leuca, distinguishing archaea and eukarya from bacteria. But how did the eukaryotic cell evolve from this prokaryotic ancestor? Part of the answer comes from looking at nucleic acid sequences. And we find, if we begin to look at, let's say, yeast and look at red algae, that some of the genes are consistent with what we find in archaea, and some are consistent with what we find in bacteria. That's true for both yeast and for red algae. The others are unique. Interestingly enough, the eukaryotic genome typically has an archaea-like set of genes for DNA and RNA processing, and bacterial genes for most of the metabolic functions. So what are the hypotheses for the origins of eukaryotes? One possibility is that an archaea and a bacteria were engulfed by a third cell, and the result being a eukaryote. In order for this to happen, engulfment would be a requirement. And recall that archaea and bacteria have cell walls, meaning that this host cell would have evolved into a form that did not have a cell wall. There are a number of contrasting hypotheses for the evolution of eukaryotic cells. In some, we have a differentiation and then an engulfment. In others, we have the engulfment and then the differentiation. I don't want to spend much time on this because at this point, we don't know which of these competing hypotheses are correct. What is most likely the important take-home message is that this engulfment was a one-time event. Just like the one-time event of cyanobacteria developing oxidative photosynthesis, this symbiotic relationship, the long-term relationship between the archaea and the bacteria into this new cell, is likely a one-time event. So keep in mind that eukaryotes are distinctive because they have this capacity to engulf things. Here you see a picture of an amoeba capturing through phagocytosis a particle which then gets incorporated. Remember that endocytosis might have been a key cellular function that defined the domain eukarya early on, allowing subsequent acquisitions. Because as I had just mentioned, archaea and bacteria have cell walls. It's not possible for them to engulf. The engulfment of a bacteria by an ancient eukaryote is shown here in this graphic. Here we have the lineage, the evolutionary tree for bacteria, for archaea, and eukarya. We believe that it is possible, and we don't have evidence one way or the other, that it is possible that the eukarya developed, and there might have been a history, maybe even a long history, where the proto-eukarya didn't have mitochondria. If that form existed, and if that form existed for an unknown period of time, there are no remnants left for us to begin to explore. All eukarya today have mitochondria without exception, or a remnant of mitochondria. So we have no part of this family, this tree of life, that we can explore. But we do have solid evidence that mitochondria came from bacteria and once were incorporated in what appears to be a one-time event. If we now look at the eukarya that were present after the end of Snowball Earth 1, we find that they all had chloroplasts, or the plants and algae had chloroplasts. They all had a membrane-bound nucleus, and they all contained mitochondria. Now, as I mentioned, the concepts of these endosymbiosis were initially meant by skepticism from a very conservative scientific community. And the hero of the story is none other than Lynn Margulis. When I met her in the 1980s, she was just as feisty as people had told me she would be. She had to be feisty. She had to argue in advance of the time when we had the nucleic acid evidence to support her hypothesis that this symbiotic relationship between a chloroplast and a mitochondria in a cell evolved. Unfortunately, received so much criticism, but she was a tough woman, and she held her ground. Today, Lynn Margulis' name is as synonymous with symbiosis as Charles Darwin's name is for the concept of evolution. Thank you, Lynn. Now, in the symbiosis theory, it's important to note that the mitochondria and the chloroplast each have their own DNA. Not all of the DNA for the functioning of oxidative phosphorylation or photosynthesis comes from nuclear DNA. In part, it's from both. So where did the mitochondria come from? The genetic evidence today suggests that the mitochondria came from a small group in the Rickettsidea called the proto-mitochondrian group. Those are the closest relatives in terms of nucleic acid sequences. So it's in this protobacteria that the mitochondria came from. Interesting, if you begin to see what else is in this group. It's a group of parasites that often cause diseases, such as typhus, giving us typhus fever that can be deadly if not treated.